So hello everyone, I'm Amita, and in this session we will be discussing a very fundamental topic related to clinical genetics, which is autosomal dominant inheritance. So before going into autosomal dominant inheritance and what are its characteristics, let's just give you a brief overview of genetic disorders. So if we look at the etiology, genetic disorders can be classified broadly into chromosomal disorders, which are caused by abnormalities in chromosomes, single gene or monogenic disorders, which are due to defects in a single gene, and polygenic or multifactorial disorders, which are caused due to defects caused by both genetic and environmental factors. So today we will be focusing on monogenic disorders. These disorders are also called as Mendelian disorders because they follow characteristic patterns of inheritance in families, which are in accordance with Mendel's law of inheritance. So autosomal dominant inheritance follows Mendelian principles of dominance of a particular trait or characteristic over the other traits. The principle of segregation, where each of the trait has segregates independently and also independent assortment. So these are disorders which are caused by genes which lie in the autosomes. And how do we determine what if a particular disorder follows autosomal dominant inheritance is from the pedigree, which looks something like this, which I'll be explaining in the subsequent slides. So the characteristics of autosomal dominant inheritance are where only a copy of the abnormal gene is required to produce the phenotype. So we all know that for each gene, we have two copies or two alleles, one inherited from each parent. And when the when the genotype is different at the locus in both the alleles, the individual is said to be a heterozygote for that particular genotype. So in autosomal dominant inheritance, one abnormal copy is enough to produce the phenotype. If we look at the pedigree, multiple generations are affected. As you can see here, all the three generations are affected, which are denoted by the filled circles, which are affected females, and the squares, filled squares are the affected males. So both males and females are affected almost equally, and both males and females can transmit the disease. So the female has transmitted the disease in this particular condition, and the male has transmitted the disease here in this family. And the most important characteristic which actually distinguishes this type of inheritance from X-linked dominant inheritance is a male to male transmission. So when there is a male to male transmission, it distinguishes a autosomal dominant pedigree from a X-linked pedigree. So the autosomal dominant disorders, as we have already discussed, the phenotype is expressed in the heterozygotes. But if we look at the homozygotes here, where the genotype is same at a particular locus in both the alleles, the phenotype is also expressed in the homozygotes as well as in the heterozygotes. So depending on the extent to which the phenotype is expressed, such disorders are classified into pure dominant autosomal disorders or incompletely dominant autosomal disorders. So in pure dominant disorders, the phenotype is same as in the heterozygotes as well as in the homozygotes. Whereas in incompletely dominant, or sometimes called as semi-dominant disorders, the homozygotes are more severely affected as compared to the heterozygotes. Then we have some disorders where the phenotypic expression of both the alleles is co-dominant. That is, both the alleles express equally. One is not dominant over the other, so it is not masked by one of the alleles. So both the alleles express equally, and such type of disorders are said to follow co-dominance. A particular example is of the ABO blood group, where an individual with AB blood group expresses both A and B antigens. So if we look at the autosomal dominant disorders that we have, most of them, most or almost all of them exhibit semi-dominance. Pure autosomal dominant disorders are very, very rare, if at all it exists. Some of the examples that we commonly see of autosomal dominant disorders are listed here. Achondroplasia, which is caused by FGFR3 mutations. So we all know that it is because of heterozygous mutations. However, homozygote individuals also do have achondroplasia, but of the severe variety. So it is said to exhibit semi-dominance. So also in adult polycystic kidney disease, as we have yet to actually characterize individuals with homozygous mutations in PKD1 or 2 gene. Marfan syndrome, again, the homozygotes in FBN1 gene are said to exhibit more severe phenotype as compared to the commonly seen heterozygotes. Then we have familial hypercholesterolemia, which also follows similar pattern, hereditary breast and ovarian cancer. And Huntington disease is one disease where it was believed to follow pure dominance. However, in recent times, it has been seen that individuals who have mutations in both the alleles, that is homozygotes or compound heterozygotes, they exhibit a more severe or earlier onset of the disease, thereby showing that it also has semi-dominance. So I'll try to explain this more with the help of an illustrative case here. This is a family who presented to us with the proband at 5 years, a female who had this kind of skin lesions, which are xanthomas over the joints and the extremities, and also had xanthelasma over the eyelids. Her lipid profile showed hypercholesterolemia, and thereby a clinical diagnosis of familial hypercholesterolemia was made. If we look at the family history, we have the father who has some form of heart disease, mother who was apparently healthy, and one elder brother who passed away at the age of 17 years because of some heart disease, which was not conclusively identified. So when we tested this particular proband, she was found to have a homozygous mutation in the LDLR gene, and reviewing the records of the other family members and the genotype, based on the genotype test, conclude it was this was the segregation or the uh, explanation of the disease in the family at the gene level. So here we have the proband who has both the alleles of the LDLR gene mutated as shown by this cross marks here, and the mother and the father, they have one copy which is mutated, one copy is unaffected or wild type as shown here, whereas one copy is mutated, so they are heterozygotes for the same mutation. So the father also has a high LDL level, a high cholesterol level, as similarly the mother also has xanthelasma, which was not picked up and initially, and also had a lipid profile suggestive of hypercholesterolemia. The brother who passed away was also most probably having the homozygous form of the disease, as evidenced by his lipid profile. The sister who was apparently asymptomatic was not tested, and these are the possible genotypes. Either she has inherited both the wild copies from the parents, in which case she will be unaffected, or she might be a carrier of mutation in one of the alleles, that is, she is a heterozygote. She does not have any symptoms at 11 years of age, so she is unlikely to be a homozygote for the said mutation. So this is a family which illustrates the segregation of an autosomal dominant trait, that is familial hypercholesterolemia, which shows phenotypic expression in heterozygotes and a more severe expression in homozygotes, thereby showing incomplete dominance. So what is the recurrence risk in such families? So the risk of recurrence and the severity depends on either one of the parents is affected or both parents are affected, or whether the trait is a pure dominant condition or is incompletely dominant. So if we look at the example here, the red allele is the mutated allele, which is denoted by the capital A, and the other alleles are the wild type or normal alleles. So we have one parent which is a heterozygote for the mutant allele, and another parent who is normal or has both the wild type alleles. So the possible gametes uh, uh, which because of this mating are given in this Punnett square, and the possible combinations are given here. So if we have a capital A and a small a, then this individual will be a heterozygote, and the individual with both alleles having the small a, they will be wild type. So the individuals with the heterozygote here, as denoted by this colored letters, they will be affected, or phenotype expression will be seen in these individuals. Hence, out of the possible four combinations, half, two of them will be affected. So the risk of recurrence is 50% in such case. That is when one of the parents is affected and is a heterozygote. What happens when both the parents are affected? So we have an example here of a family with achondroplasia. Again, achondroplasia, heterozygote, autosomal dominant disorder. So the father is a carrier of a mutated allele here, the mother has a mutated allele here. They are heterozygotes since only one allele is affected. The possible combinations of the alleles in their offspring are denoted here. So either both the mutated alleles are inherited, in which case the individual will be homozygous for the mutation. One of the alleles may be inherited from either parents, in which they will be similar to the parents, that is heterozygotes, or an individual, the offspring might inherit the wild allele from each parent, in which he or she will be have no phenotypic expression, that is a normal individual. So this heterozygotes will have achondroplasia similar to the parents, and this homozygous individual, they are more severely affected, and they usually result in perinatal lethality. So this is again, so if we ask the risk of recurrence, it depends on what exactly the status of the parents are and what type of condition we are concerned with. Then autosomal dominant disorders also exhibit certain characteristics which are important in understanding these disorders and familial patterns of such diseases. I'll be discussing briefly each of these. So first, what is pleiotropy? Pleiotropy is when a single mutation or a single defect in a single loci leads to multiple distinct traits involving different organ systems. For example, we have von Hippel-Lindau disease, where a mutation in the VHL gene causes cerebellar hemangioblastoma, retinal angiomas, then carcinoma of the kidney, pancreatic cyst, etc. Then we also have tuberous sclerosis, where the brain is affected in the form of cortical tumors and ependymomas, hypomelanotic macules in the skin, angiolipomas, adenoma sebaceum, then rhabdomyomas in the heart, etc. So the mutation here in such conditions are single. There is only one single mutation. However, the effects can be seen in many different organ systems, leading to a multi-systemic kind of presentation. So this such conditions are said to show pleiotropic effects. So one case which will further help us in understanding these various characteristics are this case where the 10-year-old male has come to us with poorly controlled seizures and a poor scholastic performance. On examination and further investigation, he had this cortical tubers and subependymal lesions. Then he also had this hypomelanotic macules over the trunk and chagrin patch, and therefore a clinical diagnosis of tuberous sclerosis was made in this child. Then we all know that tuberous sclerosis is an autosomal dominant disorder, and many cases are inherited from the parents. So in such cases, we have to examine the parents. So in this family, examination of the parents revealed that the mother had this kind of lesions, which are the subungual fibromas in the nails. This is also a feature suggestive of tuberous sclerosis. So these phenomenon is called variable expressivity. It is the degree to which a particular phenotype is expressed in a person. So we have the same mutation here. The mother also has the same mutation as similar with the proband. However, the severity of expression is different. The mother has only mild presentation, whereas the child has a severe neurological phenotype. So this same mutation, this variable expressivity may be seen in the same family, or it may also be seen in different families. So such situations pose challenges in diagnosis, where if we don't have a detailed phenotype of the parents and all, it may help, it may lead us to incorrectly determine the risk of recurrence and also the management issues in the family. The possible etiology of why such phenomenon is seen are exactly not known. However, this is believed to be because of modifying factors, which can be genetic, the genetic background, or other genes involved in modifying the phenotype in certain individuals, as well as environmental factors. Then we have another characteristic, which is how penetrant an autosomal dominant disorder is. So penetrance is the probability that a mutant allele will have a phenotypic expression. So if we have 100 individuals with the same mutation and examine them, and out of the 100, if only 90 individuals show that particular phenotype, then we say that this particular trait or this particular condition shows 90% penetrance. Then therefore it is incompletely penetrant, since 10 individuals still do not show the phenotype in spite of having the mutation. So if we look at the pedigree here, there is autosomal dominant inheritance as evident by multiple generations being affected. There is male, female, both are affected, and there is transmission from the females. And here in this particular family, if we see, then this individual denoted by the arrow here does not show the phenotype. He is asymptomatic clinically. However, he has passed on the mutation to his son, who is showing the phenotype. So if we were to test the genotype or and undergo if this individual were to undergo the genetic testing, then he would also carry the mutation. So there is a skipping of a generation here. So this individual is affected, however, this individual is not affected, and the phenotype is again shown here in the next generation. So there's a skipping of generation, and this denotes incomplete penetrance in the family. But penetrance, we should be very cautious in actually labeling as incomplete penetrance, because it shows relativeness in terms of age of the individual. So if we were to examine the individual at a younger age, then he or she might not show the phenotype, which may be picked up later at a later age. This is especially true for adult onset diseases. And also it depends on what diagnostic parameter we are using to characterize the phenotype. If we were to only see clinical presentation of the individuals, it will show decreased penetrance. However, if we were to determine the biochemical, use some biochemical testing or parameters, then it will show that the phenotypic expression is there. So it depends on both what instrument we are using to actually determine the phenotype and what is the age of the individual when he or she is being examined. So some examples I've listed. Chondroplasia again shows 100% penetrance. So at birth only it shows the phenotype. So all individuals having the achondroplasia mutation in FGFR3 gene will express the phenotype. So this is a condition which shows 100% penetrance. Whereas on the other hand, we have hereditary ovarian and breast cancer, which is caused by BRCA1 gene. And again, with age, it is estimated that it shows only 60% penetrance by 80 years of age. That is, if we were to examine 100 individuals at 80 years of age who carry BRCA1 mutation, then out of the 100, only 60 individuals will show breast or ovarian cancer, and the rest 40 individuals will not have ovarian breast cancer despite having the particular mutation. So this is what is known by what is meant by penetrance. Then we come across in many autosomal dominant disorders, we come across such families like where the individual or the proband is the only affected member in the family. It is not inherited. None of the family members are affected similarly. So most of these conditions are caused by new mutations, which are known as de novo mutations. This is especially true for many autosomal disorders which show decreased reproductive fitness, that is, severe childhood onset diseases or lethal conditions where the affected individual does not have the fitness to pass on the mutation to the next generation. For example, we have thanatophoric dysplasia, which is perinatally lethal, and therefore all cases of thanatophoric dysplasia will be caused by new mutations and it will not be passed on from the parents. This de novo mutations have been shown to increase with the paternal age. This has been shown in certain autosomal dominant conditions like Marfan syndrome, neurofibromatosis, and achondroplasia again. So, achondroplasia, as evident by this photograph here, we all know it's an autosomal dominant with 100% penetrance. So here in this family, both the parents are normal. They do not have achondroplasia. But however, the sibling of the affected child again has achondroplasia. So how is this possible? This is explained by something which is called gonadal mosaicism or germline mosaicism, where the one of the parent is a mosaic. That is, he or she has normal cell lines as well as cell lines with the mutation in his germ cells. So either the sperm or the ova has some of the germ cells have mutation and some have normal cells. So if the mutated germ cells were to form the zygote, then the mutation will be passed on to the sibling. So this cannot be proved. There is no clinically or practical method to actually prove germline mosaicism in individuals. So in autosomal dominant disorders, in almost all of them, we can never rule gonadal mosaicism out, and hence the recurrence risk cannot be said to be zero. So in these conditions, prenatal diagnosis is still offered even in families where the mutation is apparently de novo or the proban is the only affected member in the family, since this gonadal mosaicism cannot be ruled out. And we have some adult onset disorders like Huntington disease, where an individual presents at 30 years of age. And if we take the family history, we get to know that his father also had a similar disease, but was diagnosed at a much later age of in the 50s. So also his grandfather. So here, what is striking is the phenotype is same, However, the severity and in terms of age of onset is increasing. So these individuals were diagnosed in their 50s and 60s, whereas this individual, the proband, is symptomatic at 30 years of age. So this is what is called as anticipation, where there is earlier manifestation of the disease in subsequent generations. This phenomenon is seen in a group of disorders which are caused by triplet repeat expansions, or these are also known as dynamic mutations because they tend to expand when they are inherited in subsequent generations. So some of the examples of triplet repeat disorders which show this anticipation are myotonic dystrophy, spinocerebellar ataxia. However, this phenomenon is not limited to autosomal dominant inheritance or autosomal dominant disorders. It can be seen in some recessive and X-linked disorders also. It is a characteristic of triplet repeat disorders. What is the underlying molecular mechanism of dominance in these disorders? So three main mechanisms are known to cause dominance. One is haploinsufficiency, where deficiency of half of the normal gene product or the protein is the cause of the disease. This is very commonly seen. Some examples are porphyria and neurofibromatosis type 1. Then the second mechanism is gain of function effect, where the normal protein is increased because of the increased gene dosage, thereby leading to increased function, or the new mutation may cause the protein to have an altogether new function. So example again, achondroplasia, which is caused by a single mutation in all individuals, and Charcot-Marie-Tooth disease, which is caused by duplication of PMP22 gene. Then in some conditions, we have a dominant negative effect where the abnormal protein interferes with the normal function of the protein. This is seen characteristically in diseases where the proteins are part of a multimeric complex, like the collagen related osteogenesis imperfecta caused by COL1A1 on COL1A2 genes. So these are the molecular mechanisms. So just to summarize the characteristics exhibited by autosomal dominant disorders, if we look at the pedigree, and we know that this child has an autosomal dominant disorder and both the parents are asymptomatic, what could be the possible causes in this particular family of this if we look at the genotype level? So it could be because of a new mutation in the proband, which is a de novo mutation. That is, it is not inherited. The mutation has occurred as a new occurrence only in the proband. Then it may be reduced penetrance. One of the grandparents might be affected. We don't have information regarding that, so we cannot say. So this is still a possibility. Then the third possibility is again variable expressivity, where one of the parents might show a very mild manifestation of the phenotype, which may easily be missed, as was in the case of tuberous sclerosis family. Then one of the parent might carry the mutation but have a delayed onset, which is again reduced penetrance, or it can be because of anticipation also, where the condition is a triplet repeat disorder, and the proband has earlier manifestation and the parent has not manifested it yet. Then one of the parent might be a mosaic, and we might be able to conclude that if again there is a recurrence in the sibling. So these are the possible effects, and last of all, then issue of non-paternity can still be there. So this is all. Thank you all for your attention.